Hey everyone, today's set of notes we're focusing on the concept of antigens and how your immune system can use these molecules known as antigens to help mount an immune response if needed. So we're specifically trying to address the distinction between self-antigens and non-self antigens. So by the end of this video, you should be able to tell me the difference between the two and how we can then use these in our immune system to recognize things that belong to our body versus things that are foreign or invading our body, such as a pathogen.
Antigens. So these particular molecules are usually found on the surface of cells. So all living cells have these molecules called antigens on the surface of them. As I said previously, they are usually made of proteins, but they can be made of other things. Not a huge piece of information that you need to remember, but it might help some of you with your understanding if you can make the connection that antigens can be made of proteins and what we already know about what proteins can do. So all cells have these molecules on their surface called antigens. They are specific, okay? So the antigens are specific to that particular organism. And they kind of act like a fingerprint for a cell. So just like every human being is meant to have its own unique fingerprint, cells that originate from an organism have their own unique antigens on the surface of them that allow them to be distinguished from cells of another organism. To really simplify that down, let's have a look at the diagram that's on the screen here. So a very simplified version of what an antigen is. We're going to pretend that antigens are being represented in this diagram by the colored circles. So let's pretend, for example, that this gray cell that you're seeing with the colored circles on them is an example of a cell from my body. So my body has produced this cell and it contains antigens that are pink, yellow, blue, and green in color. Those antigens that are on the surface of my cells, because my body produced them, to me, they are known as self-antigens because they belong to me. They were created by me, myself, so they are self-antigens. If a cell was to if I was to compare my cell to a cell that didn't originate from my body, so for example, a cell that came from your body, I would not expect to see the same antigens on the surface of your cell. You are not identical to me in any way. We come from different parents. Even my own siblings, they wouldn't have the same antigens on the surface of their cells as I do. Probably the only exception to the rule about having the same antigens on the surface of your cell, and again, this is not always 100% of the time, would be if you were an identical twin. So if I was to sit my cell side by side with your cell, my antigens, my self-antigens that I see on the surface of my cell are going to be very different to the ones that I see on the surface of your cell. Now, if we look at this image on the slide here, you can also see that on the other side, we have these receptor shapes being labeled. So there's receptors for self, okay, and you can see that these receptor shapes here, these ones, are complementary in shape to the colored circles that are on the surface of my cells. Those receptors are there to be able to detect one of my own cells. So in my body, this cell is going to run into other cells that belong to me, and they need to somehow recognize, hey, this cell actually belongs in this body. And they do that by having these receptors on their cells to interact with those self-antigens on another cell. They also need to have the ability to detect cells that don't have any of those self-antigens. So for example, if this particular cell was to come into contact with one of your cells, we might have some non-self receptors being used. These non-self receptors are receptors that would have a shape that is complementary, potentially complementary, to antigens that would be presented on your cells. The antigens that are presented on your cells, which are different to the antigens that are on the surface of my cells, would be classified for me as non-self antigens. Because your cells did not originate in my body, your cells, for me, contain non-self antigens. So when we're looking at a particular organism or looking at cells and identifying markers or antigens on their surface, the organism that we're focusing on, we refer to those markers or those antigens as self. And if we are then comparing an invading pathogen, which is what we're going to be looking at in immunity, the invading pathogen is the thing that has non-self antigens. So how do these antigen molecules on the surface of cells play a role in the immune system? Well, that's what your immune system, which we're going to look at a little bit more as we move along, that's what your immune system is focusing on when it interacts with a cell. And it is looking for the presence of a self-antigen. And in that case, it would identify that particular cell as self. So your immune system is very happy. Yay, you've got a self-antigen on the surface or multiple self-antigens on the surface. And your immune system won't do anything. It's recognized this cell belongs. I don't need to start defending the body. I'm perfectly happy to leave things as it is. It's when your immune system, the cells in your immune system, detect non-self antigens, so antigens that didn't originate from that body, that's when your immune system starts to take action. So they've identified these antigens as non-self. They did not originate from the body. We're assuming that they have probably originated from a pathogen, such as a bacteria or a virus. And if that is the case, your immune system needs to take action and follow a series of steps to remove or destroy that non-self antigen, potentially a pathogen, from that organism's body. Some new words that you're going to come across today. These new words that are on this slide that I'm going to point out to you are another series of terms used to describe self-antigens found on human cells. Okay, so this is for humans only. So the first series of terms that we're going to use is something called a major histocompatibility complex. You don't need to remember the full name of it, but please recognize MHC. This is a term that you're going to come across quite a bit. So MHC is another way of saying a self-antigen. We usually refer to them as MHC markers. Okay, so MHC markers are the same as saying self-antigen. And the other one that we sometimes come across but is less common, we probably won't use this too much, but just in case you come across it in your textbooks or other resources, is the human leukocyte antigen, or HLA. Okay, so again, you're not expected to remember the full name or anything like that. So it's usually referred to as HLA marker. And again, that's the same as saying self-antigen when referring to humans. And the difference between the MHC markers and the HLA markers are the HLA markers tend to only be found on blood cells, okay? Whereas your MHC markers are found on almost every cell in the body. Obviously there are exceptions, but don't stress too much about that. So as we were saying before, your combination of antigens, so you've got hundreds if not thousands of antigens on the surface of your cells, the combination of those antigens is unique to every person. So that makes sense that the combination of MHC and HLA markers on the surface of every cell will be unique to every person. No one is going to have the same combination as you. And that's it for this slide. Just touching on a little bit more information on the MHC markers, as these will come up again when we look at more detailed responses from your immune system in terms of what cells are involved and how they defend or attack pathogens and non-self antigens. So MHC markers, okay, so they are self, and they can be used to identify a cell as belonging to that particular person, but it can also be used to identify a cell as not belonging to that particular person. Two different categories that we will come across in our study of the immune system is an MHC class 1 marker. Okay, so that little line is actually representing 1, and the one over here is representing 2. Okay, think of them as Roman numerals. So an MHC class 1 marker is referring to the antigens that are found on the surface of every cell in your body except for red blood cells. Okay, so every cell in your body, regardless of what cell it is, will have MHC class 1 markers scattered all over it, except for your red blood cells. Remember, an MHC class 1 marker is a more specific name for describing an antigen, a marker on the surface of the cell that we recognize as self. We also have this category of MHC class 2 markers over here in purple or pink on your screens maybe. Now these ones are a little bit more special because they have an extra function which we will talk about in a later set of notes, but these MHC class 2 markers are not found on every cell. They are only found on specific cell types. They are found on something called antigen presenting cells, B cells, and T cells, and we will talk about what those types of cells are as we progress further through this unit. But just to be aware now so that you're getting introduced to it at the start, we have these molecules on the surface of our cells called antigens, or sometimes we can refer to them as MHC markers or MHC class 1 or MHC class 2 markers. These markers or these antigens that are on the surface of our cell, because they belong to us, they originated from our body, we refer to them as self-antigens or self-markers. We can also expose our immune systems to cells that have antigens on the surface or MHC class 1 or class 2 markers on the surface that we would classify as non-self. And we would classify those markers or those antigens as non-self if they were originating from an organism that wasn't us. So it could be something like a bacteria or potentially a virus, type of pathogen that does have non-self markers on it. But it could also be something as simple as I've had a blood transfusion because I lost a lot of blood in a car accident, and that blood that's being injected into my body wasn't created by my body, it was created by someone else's. So a lot of those blood cells are actually going to be flagged by my own immune system as containing non-self antigens. And my immune system will eventually try and destroy those blood cells that have been pumped into my body. All right, before I move on to the next slide, just want to make a really clear point here. If you're struggling to grasp the concept of what an antigen is, can you please make sure you send me an email, you send me a message on Teams, you communicate with me in some way so that I can help you with your understanding of that. Because if you are struggling to grasp the concept of an antigen, you will probably struggle to grasp the next couple of concepts that follow on in the next week or so. And I'd rather nip this in the bud now than let it blow out of control. Okay, thanks guys. All right, moving on.